Hi everyone, we are going to talk about the fatty acids in this chapter. Fatty acids are fuel molecules. They are stored as triacylglycerols, which are uncharged ester of fatty acids with glycerol. Triacylglycerols are highly concentrated stores of metabolic energy because they are reduced and anhydrous. The yield from the complete oxidation of fatty acids is about 38 kJ per gram, in contrast with about 17 kJ per gram for carbohydrates and proteins. The basis of this large difference in caloric yield is that fatty acids are much more reduced than carbohydrates or proteins. Furthermore, triacylglycerols are nonpolar, so they are stored in a nearly anhydrous form, whereas much more polar carbohydrates are more highly hydrated. Consequently, a gram of nearly anhydrous fat stores 6.75 times as much energy as a gram of hydrated glycogen, which is likely the reason that triacylglycerols rather than glycogen were selected in evolution as the major energy reservoir. In mammals, the major site of triacylglycerol accumulation is the cytoplasm of adipose cells. The fuel-rich tissue is located throughout the body, notably under the skin and surrounding the internal organs. Droplets of triacylglycerol coalesce to form a large bubble, so-called a lipid droplet, which may occupy most of the cell volume. Adipose cells are sphericalized for the synthesis and storage for triacylglycerols and for their mobilization into fuel molecules that are transported to other tissues by the blood. So the unity of triacylglycerols as an energy source is dramatically illustrated by the abilities of migratory birds, like the American golden plover flies from Alaska to the south tip of South America. A large segment of flight, it's about 3,800 kilometers or 2,400 miles. It's over open ocean, so where the birds cannot feed. Most lipids are ingested in the form of triacylglycerols and must be degraded to fatty acids for absorption across the intestinal equilibrium. Intestinal enzymes called lipases, secreted by the pancreas, degrade triacylglycerols to free fatty acids and monoacylglycerol. In the intestinal mucosal cells, the triacylglycerols are resynthesized from fatty acids and monoacylglycerols and then packaged into lipoprotein transport particles called chylomicrons. So it's a stable particles about 2,000 angstrom. It's about 200 nanometer in diameter. These particles are composed mainly of triacylglycerols, with apolipoprotein B48 as the main protein component. Protein constituents of lipoprotein particles are called apolipoproteins. Chylomicrons also transport fat-soluble vitamins and cholesterols. The chylomicrones are released from the lymph system and then into the blood. Fatty acid degradation and synthesis consists of 4 steps that are the reverse of each other in their basic chemistry. Degradation is an oxidative process that converts a fatty acid into a set of activated acetyl units we called acetyl-CoA that can be processed by the citric acid cycle. An activated fatty acid is oxidized to introduce a double bond. The double bond is hydrated to introduce a hydroxy group. The alcohol is oxidized to a ketone. And finally, the fatty acid is cleaved by coenzyme A to yield acetyl-CoA and a fatty acid chain 2 carbons shorter. If the fatty acid has an even number of carbon atoms and is saturated, the process is simply repeated until the fatty acid is completely converted into acetyl-CoA units. Fatty acid synthesis is essentially the reverse of this process. Tissues throughout the body gain access to the lipid energy reserves stored in adipose tissue through 3 stages of processing. First, the lipids must be mobilized. In this process, triacylglycerols are degraded to fatty acids and glycerol, which are released from the adipose tissue and transported to the energy-requiring tissues. Second, at these tissues, the fatty acids must be activated and transported into mitochondria for degradation. Third, the fatty acids are broken down in a step-by-step fashion into acetyl-CoA, which is then processed in the citric acid cycle. Before fats can be used as fuels, the triglycerol storage form must be hydrolyzed to yield isolated fatty acids. In adipose tissue, these hormones glycogen and epinephrine trigger 7-HTM receptors that activate adenylacetyl cyclase. The increased level of cyclic AMP then stimulates protein kinase A, which phosphorylates 2 key proteins, paralipin, a fat droplet-associated protein, and hormone-sensitive lipase. The phosphorylation of paralipin triggers the release of a co-activator for adipose triglycerol lipase, short for ATGL. Once bound to the co-activator, ATGL initiates the mobilization of triglycerols by releasing a fatty acid from triglycerol, forming diacylglycerol. Diacylglycerol is converted into a free fatty acid and monoacylglycerol by the hormone-sensitive lipase. Finally, a monoacylglycerol lipase completed the mobilization of fatty acids with the production of a free fatty acid and glycerol. So thus, the epinephrine and the glycogen induce lipolysis. In order to reach tissues that require fatty acids, the released fatty acids bind to the blood protein albumin, which delivers them to tissues in need of fuel. Glycerol formed by lipolysis is absorbed by the liver and phosphorylated. It is then oxidized to dihydroxyacetone phosphate, which is isomer to glyceraldehyde 3-phosphate. This molecule is an intermediate in both the glycolytic and the glycogenetic pathways. Hence, glycerol can be converted into pyruvate or glucose in the liver. The reverse process can take place by the reduction of dihydroxyacetone phosphate to glycerol 3-phosphate. Hydrolysis by phosphate then gives glycerol. So thus, glycerol and glycolytic intermediates are readily interconvertible. Fatty acids separate from the albumin in the bloodstream and diffuse across the cell membrane with the assistance of a transport protein. In the cell, fatty acids are shuttled about in association with fatty acid binding proteins. Fatty acid oxidation occurs in the mitochondrial matrix, but in order to enter the mitochondria, the fatty acids are first activated through the formation of a thioester linkage to enzyme A. Adenosine triphosphate ATP drives the formation of the thioester linkage between the carboxy group of fatty acid and the sulfur hydride group of coenzyme A. This activation reaction takes place on the outer mitochondrial membrane, where it is catalyzed by acetyl-CoA synthesis. Fatty acids are activated on the outer mitochondrial membrane, whereas they are oxidized in the mitochondrial matrix. A special transport mechanism is needed to carry activated long-chain fatty acids across the inner mitochondrial membrane. These fatty acids must be conjugated to carnitine. The acid group is transferred from the sulfur atom of coenzyme A to the hydroxy group of carnitine to form acid carnitine. The reaction is catalyzed by carnitine acid transferase A, also we called carnitine pymalitosyltransferase 1, which is bound to the outer mitochondrial membrane. Acid carnitine is then shuttled across the inner mitochondrial membrane by a translocase. The acid group is transferred back to coenzyme A on the matrix side of the membrane. This reaction, which is catalyzed by carnitine acid transferase 2, is simply the reverse of the reaction that takes place in the cytoplasm. A saturated acetyl-CoA is degraded by a recurring sequence of 4 reactions. So, oxidation by flavin-adenine dinucleotide, short for FAD, hydration, oxidation by NAD+, and thiolysis by coenzyme A. The fatty acid chain is shortened by 2 carbon atoms. As a result of these reactions, and FADH2, NADH, and acetyl-CoA are generated. Because oxidation takes place at the beta carbon atom, this series of reactions is called the beta oxidation pathway. Okay, so that's all for today. Thank you for your listening.